At Amphoraxe, we develop drugs to replace ineffective or restricted antibiotics. Every year, the problem of increased antibiotic resistance is claiming millions of lives across the world. To curb this trend, regulators have been limiting antibiotics use in farms. But the new regulations also increase animal mortality and morbidity and reduce productivity and food safety. To solve this problem, we are developing antimicrobial peptides as antibiotic replacements. AMPs are small protein fragments, structurally and mechanistically different from antibiotics. They kill faster, have broad spectra, and do not interact with the DNA of their target pathogens. As a result, they do not create superbugs. Partnering with an academic consortium, we are using genomics and bioinformatics methods to discover and develop novel AMP-based drugs. Our studies have reached a technology readiness level of 4, where we have validated our discoveries in a laboratory environment. The table shows an excerpt of our test results against a set of bacteria, including their multi-drug resistant varieties. We have also tested our AMPs for their toxicity profiles against the animal blood. Low numbers on the table at the top and high numbers on the table at the bottom indicate better performance. The table also demonstrates how our designs based on original discoveries improve performance and lower toxicity. We expect to push a list of our most successful AMP designs for animal experiments starting January next year.